Gene Expression Clustering Tool Genomic Data Commons, GDC 2.0 Video Guides, Charlotte Sackett, GDC User Services Scientific Support Analyst, Center for Translational Data Science, University of Chicago. Introduction to the Gene Expression Clustering Tool. The GDC's Gene Expression Clustering Tool can be used to visualize the top most variably expressed genes in a cohort. How to access the Gene Expression Clustering Tool. To access the Gene Expression Clustering Tool, we will first load the GDC's Data Portal homepage at portal.gdc.cancer.gov, then click on Analysis Center, which is the computer screen icon in the upper left corner of the web page. Under the Analysis Tools section, we will locate the Gene Expression Clustering card, which is fourth in this tutorial. Please note that the locations of the tool cards may change, so it is important to read the title of the cards. Once we have located the Gene Expression Clustering card, we will click on the Play button in the upper right corner of the tile. How to use the Gene Expression Clustering Tool. The Gene Expression Clustering Tool displays the most variably expressed genes, which are selected from the 738 cancer census genes for the first 1,000 cases in the current cohort. If the active cohort is not of interest for this analysis, we can switch cohorts by clicking on the drop-down menu and selecting a different one at any time. Note that before we can run the Gene Expression Clustering Tool, one cohort must already be created. If you have not created a cohort yet, please pause this video and use the Cohort Builder tool to create one. You can refer to the GDC Cohort Builder video for assistance. This tutorial will be demonstrated with a previously created cohort of 50-plus-year-old lung cancer cases with RNA-seq data. The Gene Expression Clustering Tool displays a heat map visual with the active cohort's cases above, a column of genes on the left, and the gene expression value ranging from -5 to +5. Hovering over a coordinate in the grid displays the case ID, gene, and gene expression value. This particular coordinate for case TCGA858071 and gene ACKR3 has a gene expression value of approximately 5.48. Several gray buttons which appear above the graphic can be used to customize the visual. The clustering button allows users to select whether they want to use an average or a complete clustering method. We can also change the column dendrogram height, the row dendrogram width, and the z-score cap from this button. To the right of the clustering button is another one titled Number Cases, which can be used to specify the maximum length of case labels. Next, the Number Genes button offers various ways to display how many cases are represented for each gene, including absolute, percent, and none. We can also alter the row group and row label maximum lengths, as well as choose whether we want to have a stacked or oncoprint rendering style. We can also sort genes by input data order or by case count. The last option in the Genes button allows us to edit and create new groups. By default, there are 100 genes displayed for our cohort. Clicking on the gray Edit Group button displays all 100 genes, each of which can be removed by clicking on the red X in the upper right corner of the gene's corresponding button. Let's remove the ELF3 gene by clicking on the red X within its button. We can also edit this group by adding top variably expressed genes. Let's load the list. Now we can see 4 rows of genes. For these changes to reflect in the visual, we need to click the gray Submit button below the list of genes. The top 50 most variably expressed genes now appear in the visual. Notice that the Number Genes button now displays 50 instead of 100. Let's click on the Number Genes button again. We can also create a new group, which requires a name. We will enter Test Group into the text box labeled Group Name. To add genes to our group, we can either search for a gene in the text box, choose to load top mutated genes, or load msigdb gene sets. Let's add ELF3 to this gene set by searching for it in the text box. Note how we see a green check mark indicating that we have entered an accepted gene. Instead of searching for individual genes using the text box, we can also choose to load top mutated genes. Alternatively, our third option is to load an msigdb gene set, which will display groups H and C1 through C8. Let's expand the ontology gene sets by clicking on the plus sign next to C5, expand the Go Gene Ontology gene sets, expand the BP subset of Go, then select the first gene set GOBP_10_4-mel-tetrahydrofolate_metabolic_process. This will load the associated genes, which we can save by clicking the gray Submit button below. The gene expression clustering visual will now refresh to reflect these changes. Note that if there are no gene expression values for the group of genes and our cohort, the visual will appear blank in that section. The Variables button can be used to add specific variables to the grid, which are grouped into 7 categories: demographic, diagnoses, exposures, family histories, project, samples, and tissue source site. Other variables that do not fall into those 7 categories also appear in the list. We can add gender to the visual by clicking on the plus sign to expand the demographic category and selecting the blue gender button. Once it turns yellow and displays a green check mark to the right of it, both of which indicate that it has been selected, we can click the gray button labeled Submit One Term, and gender will appear at the bottom of the graphic. Hovering our cursor over the gender row shows the gender for each corresponding case ID in the grid. In this example, I have stopped over case TCGA 928064, who is a male. The next button, called Cell Layout, allows users to modify the format of the cells by changing the colors, cell height, width and spacing, and label formatting. Similarly, we can use the Legend Layout button to change the font size, height, width, and other formatting preferences for the legend. Finally, the Download button will download the matrix in SVG format. To the right of these gray buttons is an option to zoom into the visual. This is set at 1.0 by default. We can use the up and down arrows or the sliding scale to zoom into or out of the graphic. Let's zoom in as much as possible. Now we can start to make out the case IDs that appear at the top of the visual. Clicking on a case opens a pop-up box with a link to the GDC case summary page. I've just opened the case summary page for TCGA 734670, which we can see here. When hovering over the zoom option, a reset button appears which would cause the visual to return to its original zoom value of 1. We can also click on the case's dendrogram to select a chunk of cases. Once we click on a branch, the cases in that selection are highlighted with red, and we can see a pop-up window giving us the options to zoom in, list the cases, and create a cohort. Let's first zoom in, then we can use the slider function to zoom in further such that we can see the case IDs. Next, let's click List Number Cases, which opens a pop-up where we can see all of the cases with hyperlinks to the case summary page for each. Finally, we can create a cohort using the third option. We'll click Create Cohort, then name it Test Cohort. Once we click Save, we will see a message indicating that our new cohort has been saved. Next to the zoom option are 2 icons which can be used to switch the display of the cursor between an arrow and a hand symbol. Furthest to the right are 3 buttons labeled Undo, Redo, and Restore, which can be used to undo changes, redo changes, and restore the graphic to its default state. In the left-hand column of genes, each can be clicked on to open a pop-up window where we can display the protein paint lollipop and the GDC gene summary page. Let's click on the gene 6.1 and launch the lollipop by clicking on its gray button. The protein paint lollipop displays a lollipop chart panel and legend panel. The lollipop chart panel shows the protein view for the default isoform, including the nucleotides, codons in the exon region, introns, and protein domains. The legend panel contains the protein domains with links to databases such as the Conserved Domain Database, CDD, and PFAM. Below the legend panel is a visual displaying GDC mutation classes for the variants. For more information on the lollipop, please refer to the GDC documentation site or the protein paint tutorial, which can be found in the GDC 2.0 video guides. We can close the lollipop by clicking the X in the upper left corner of the visual. Next, we can launch the gene summary page for 6.1 by clicking on the gene, then selecting the link to gene summary. This opens a new tab with the 6.1 gene summary page. Our last option is to remove a gene from the visual. Let's click on the 6.1 gene, then select the Remove option. We can see that the 6.1 gene no longer appears in the tool. Exiting the Gene Expression Clustering tool. To exit, we can click the X button to the left side of the Gene Expression Clustering header, which will bring us back to the Analysis Center. For more guidance on this and other GDC data portal tools, please refer to the video tutorials or go to docs.gdc.cancer.gov.